Hello, Anatomy and Physiology. It is Mrs. Lloyd with you here virtually from home. I am feeling much better, although I won't be able to be back to school until Monday. So I thought I would come in and do your lecture with you guys this way today. And so this video will also be made available for the students who are at home right now, and so they can pop in on the video lecture as well. Also, if you ever want to go back and rewatch this at any point, I'll post this in with our Unit 2 notes, so you can go back and get the material that way as well. I'll also share this slideshow that I created to use for the presentation as well in Google Classroom, so if I move too fast or you missed something, you can go back and get access to it that way. Also, if at any point you need to ask questions or go back and I move too fast or something and you need to write something down, just raise your hand and ask your guest teacher, and she can pause the video and go back if need be. I am here at home and my dogs are around. Penny and Norm are just on the other side of this screen. If you hear any snoring or noises, speaking of the devil, here's Rufus coming in to say hello. So I have my own audience behind the screen here. In fact, I'll show you. Let me get a little view here. We got Rufus, Penny, and Norm joining in here for our lesson on the macromolecules. So we're going to be in your notes on pages 2, 3, and 4 today. And to kind of keep things nice and organized, the first thing I want you to do is to label each page based on the macromolecules that we're going to be looking at. So this page I'm looking at right here is page 2. I want you to label at the very top of that page proteins, and then go ahead and put the monomer are amino acids there at the top. We'll come back and spend some time on this. On page 3 here, we have the carbohydrates, and so we will be looking at those and talking about our carbohydrates. Now, also on this page, halfway, this page is shared with also the lipids. And so we're going to either draw a line or you can box around these pictures that are the lipids. We're going to be labeling some on this picture and maybe just like a word right above here. So when you draw your line, kind of be careful to give space on both sides of things. So I drew a little box here around this and called it a lipids. So anyways, these are the 3 main things we're going to talk about today, but the next page on page 4 is going to be your nucleic acids. And so go ahead and label that at the top of your page. Hi, their monomer are the nucleotides. All right. Hi, so now that we have them sort of organized, go ahead and jump back to page 2, and we're going to jump in and start talking about our proteins. So we have here what's pictured up above are the amino acids. And our amino acids, there are actually 20 of them in our body. You do not need to write all of this down. You do not need to memorize all of it. It is something in organic chemistry that is asked usually of students to memorize the amino acids and their structural arrangement, meaning memorizing not just their formula, but how all of these are arranged, which is lots of fun. It's not. It's actually why a lot of people sort of complain about organic chemistry. Is there's a lot of memorization of the molecules. But I wanted to show you that there are 20 of them, and your activity at the end of today is going to be, each of you will be assigned one to do some research on. Now, one thing I want to point out is that if you notice, all of them sort of have the same kind of similar grouping here that's in the beige, but then this piece in blue is different and unique for all of them. And so that's what gives each amino acid its own unique characteristics, as you will find out as you are doing some research on them today. All right, so on this page, you don't have much room, but you do have a little bit of space kind of underneath this picture to sort of draw these 5 squares that we're going to put on here. And so in these 5 squares, we're going to represent what is going on in each of these molecules here, because each piece has a specific name. Now, every single one of these amino acids first starts off with a central carbon, right? And so you can see that every single one of these here, they actually have their own central carbon. So in that middle box, I want you to write central carbon. Coming off of each of those central carbons, on this picture coming in from the bottom, right here, we have a hydrogen. Every single one of these has a hydrogen molecule coming off of the bottom. And then to the either side of these, you have coming in what's right here, the NH2, and then we have a COOH. The NH2 is called the amino group, and the COOH is called the carboxyl group. All of these, the central carbon, the hydrogen, the amino group, and the carboxyl group are all the same for every single one of the amino acids. All 20 of them have that same base foundation. The part that differs is the thing in green that you see on all of them up above, and we call that the variable group because it varies between each of them. It's a variable. So this picture right here shows sort of a generic, it's not a specific one, but a generic amino acid. And you can see labeled on here, we have the amino group, okay, the NH2, and then we have our carboxyl group. And the carboxyl group is actually an acid, and this is where the name amino acid comes from, the amino group, and then the acid group, the carboxyl group. Every single amino acid has those two, what we call functional groups on them, and so that's where their name comes from. And then they're all surrounding and touching a carbon and hydrogen. When I get back, we're going to build the amino acids you guys are researching with 3-dimensional molecules, and hopefully you'll be able to see that in action, basically. Now, all of the rest of these are specific amino acids with specific names, and they have unique structures and characteristics to them. The first one is the very, very easiest one. Once you have that kind of base memorized, the only thing that's on the next molecule is a hydrogen. Glycine only has a hydrogen as its variable group. It's the easiest one to memorize out of all of them. Then we have, the next one is our acid, right? Aspartic acid, and it has a carboxyl group on it, COOH, just like over here, COOH, our carboxyl group, which makes this amino acid more acidic. And so it has a characteristic of being more acidic, and that's a unique property to it. Now, the next one we have is lysine. And lysine has this long chain of carbon surrounded by hydrogen, but attached to it on the end is this NH2, which should look familiar from over here. That is the amino group. And I didn't mention this before, but the amino group, the functional group, acts kind of like a base, meaning that it accepts hydrogens, whereas the carboxyl group tends to give them up. Sometimes, by the way, you end up seeing, instead of COOH, you might see COO. That just means that carboxyl group has donated its hydrogen ion because it's acidic. So you might see a COO negative, and it's just showing it kind of in its more natural state after it's donated a hydrogen ion, typically in water. So this characteristic gives aspartic acid its acidic nature. Lysine has sort of a base behavior. And then we have cysteine. And cysteine has this sulfahydryl molecule on the end of it. and it ends up making it a polar molecule. So it behaves in a polar manner, meaning it's attracted to other polar molecules. It might dissociate in water. It's attracted to water molecules as well. So all of the amino acids, all 20 of them, get grouped into different categories. And you might even see this a little bit on the picture, but some are described as being nonpolar or polar. And then they're also described as being having a charged group. They might be basic, they might be acidic, or polar or nonpolar. You'll be learning some of this depending on the amino acid that you research. And so that's good information just to know going into that. So this is actually right here. Do you see where we have our central carbon, our base hydrogen, and then our NH2, but you see it's an NH3. And then this would be the COOH, but it's missing its H. So this carboxyl group has actually lost its hydrogen ion. It's acidic. And the NH2 is a base. It accepts hydrogens, and it's picked up the hydrogen. So it has a negative charge and a positive charge here. This is kind of how you see it in nature. So if you ever see amino acids represented this way instead of an NH2 and a COOH, it's the same thing. Basically, this H moved over here. Okay, so you might see it represented both ways, still the same type of molecule. So with all of these, we sort of describe these as being branches coming off, and then sometimes they form rings. But I do want to point out this one right here. It's pretty unique. It is, in case any of you guys are researching it, proline. And this one is unique because its chain branches off, but then it wraps around and it attaches to the amino group. All the other ones, your amino group is just kind of left hanging here. But in this case, the chain comes off and it loops back around, making it pretty unique. So if you get assigned this amino acid, when you build its three-dimensional structure, it makes it a little bit different from the others. Why this is important is because when we build proteins using amino acids, the order that these proteins go in determine the shape that that protein forms. And so proteins end up folding in on themselves. They might twist or they might clump together, or certain parts, maybe two nonpolar parts, might be unattracted to each other. And so they sort of separate in certain areas where they don't want to touch. Other areas do want to touch. And so it causes this unique three-dimensional folding of our protein structure, which you might remember from Bio 1-2. I'll come right back to this, but this picture here shows some of that. We've got some that are more like twisted rods, and then we have others that are folded in on itself. And it all has to do with the arrangement of those amino acids. And the thing that determines that arrangement of amino acids is your DNA. Now, going back to this picture right here, this video, in just a minute, but not yet, I'll have your, don't want to play it, I'll have your guest teacher pause my video and instead play this. You do not have to have all this information memorized, but it goes into the different ways that our amino acids are classified. This is going to be helpful for when you're doing your research on your amino acids, and you can sort of understand, okay, if it's describing my amino acid as being polar or nonpolar or acidic or basic, that's what you're getting at. So go ahead and pause my video now. Go ahead and watch this video in class or at home, and then we'll jump back into the rest of the proteins. Okay, hopefully you were able to watch that video, and again, you don't have to have that material memorized, but it will be hopefully helpful as you do your little research on your assigned amino acid. So down at the bottom here of page 2, we've got two pictures of different types of proteins. So I will have you guys go ahead and sort of write off to the side here protein types. They're sort of categorized in two main ways. But in general, let's talk about where we find proteins. You probably know foods that are high in protein. If you wanted to eat a high protein diet, you're working out, you want to make big, strong muscles, those things are typically found in meats that we eat because muscle has lots of protein in it. Cheese, the egg whites particularly, yolks are typically fat. But also inside of our cell, we find them just free floating around in our cell's cytoplasm. And so our cells make proteins all the time. That's what DNA's job is. And ribosomes go ahead and produce proteins, protein synthesis in our cells. And a lot of those proteins end up being formed and get put out into our cell's cytoplasm, where they float around and get used in particular ways. Depends what your body needs them for. So as I said, we group them in two main ways. We have structural or fibrous proteins. Again, anatomy giving two names for the same thing. I'll usually refer to them as structural proteins, but sometimes they're called fibrous because they are fibrous. They're sort of long and stringy, sort of how you describe what they look like. Things that we find this in is things like collagen. It gives firmness and toughness to your skin. Among other tissues that we'll learn about. Keratin, we find that in our hair, our nails, and it's also a very protective structure in our skin as well as elastin, which is sort of a elastic, a stretchy fiber that we find in a lot of our tissues, tendons, and other things that we'll talk about when we get into our tissues unit. So those are the structural or fibrous proteins. The other type are called functional proteins because they do a job, or sometimes globular. And much like the fibrous proteins look like long and stringy fibers, the globular proteins look like a big blob. But the reason why they're called functional proteins is that blob, that shape, has a very special job. And you might notice in both of these pictures, these little swirls back and forth. This is basically trying to show you like a ribbon of a whole bunch of amino acids all hooked together. So you'd have an amino acid, an amino acid, an amino acid, an amino acid, and they're all connected to each other. And depending on that order of amino acid determines these twists and these turns and these loops, all because of those unique characteristics. And maybe they're polar or an acid or a base, nonpolar, they're all going to react different ways. So some examples of our functional or globular proteins, hemoglobin, which I think that's what's being pictured in this picture here. And its job is to carry oxygen. So its glob, its shape, determines its function, just like anatomy and physiology, structure and function. In this case, the shape or the structure of the blob is going to determine its important function. In the case of hemoglobin, it's going to carry important oxygen molecules in our blood. Enzymes, which are very important, that they have a very specific shape. They sort of act like a lock and a key for our chemical reactions in our body. And then antibodies as well, which act as special markers that our cells use to identify friend versus foe inside of our bodies. Now I want to talk about enzymes specifically here, and we're going to get into them in even more detail in a minute. But some examples, we have carbonic anhydrase, catalase, oxidase, helicase, maltase. Do you see a trend here? They all end in ace. If you ever see a science word and it ends in ace, you can almost always know that it is an enzyme. Enzymes almost always end in ace, and lots of times they have the same prefix of the thing that they work on. Helicase, for example, is the enzyme that breaks apart the helix in DNA. It's what unzips your DNA when it's time for DNA replication. Maltase is the enzyme that breaks down maltose, which is a disaccharide, so two sugars, and it's going to break it apart into separate monosaccharides. And then carbonic anhydrase, This one we will talk about when we get to our blood unit. But it is the enzyme that takes carbon dioxide and hooks it to water, and it creates carbonic acid. This is the form in which carbon dioxide travels through our bloodstream, and then it goes to our lungs where we exhale. That it releases, we do the reverse reaction in our lungs. The carbon dioxide detaches from the water, and we exhale the carbon dioxide and keep the water in our body. Just writing that formula down because you'll see it again later, and so I always like to do callbacks to things that we've already seen in anatomy because it's all tied into each other, just so you see where this is going to play out this year. All right, so in the middle of this page, I believe we're still on page 2, you have this picture of this purple kind of bean-shaped structure. And this purple structure here is representing an enzyme. And an enzyme is defined as an organic catalyst that speeds up chemical reactions. A catalyst definition literally means speeds up chemical reactions. So if you're in chemistry class and you're heating up your chemical reaction, that's a catalyst. Heat is a catalyst. It just speeds it up. But to say it's organic means that it contains carbon and hydrogen, because that's what organic molecules contain. And it is so important in the human body and all living things, because we wouldn't be able to do metabolism, our body's chemistry, without it. So it's super essential. It's also very important that it maintains its unique shape, because its shape is essential for what molecules it's either putting together or breaking apart. In this example, we're putting together 2 molecules. So we have something called the active site. It's the little section here with the 2 cup holders, if you will. It's sort of like the little nests in which the substrate, that's the word we use for the thing that attaches to the active site. It's where the substrate and the active sites come together. So those 2 things are going to, whatever it is, let's say, I don't know, this is a specific enzyme hooking together 2 sugar molecules. It is just the right size and shape to fit in these substrates, and only these substrates. Just like only the right key and the right shape of that key is going to unlock the right door. You might have multiple sets of keys or multiple sets of the same locks on your house, for example, but just those 2 things are the only 2 things that fit together. And enzymes are special in that they only work with one type of substrate. And their shape is important, and this is why we don't want to denature our proteins, which is why keeping our body temperature at a nice stable level is important as well. So what happens is the substrate's going to nestle into those little active sites there, and then the enzyme is going to change its shape. Notice my kidney bean has sort of changed its little globular shape a little bit. It kind of them together, just like when you twist the key in the lock, okay? It activates something in there. The enzyme itself gives it a little twist with the addition of energy. And through this process, we actually end up removing a water molecule. We end up stripping a hydrogen and a hydroxide, an OH, off of one of each of the molecules. So 2 H's and an O make H2O. And that process is called dehydration synthesis. That's a key vocab word, very important that you pick up on today. Dehydration synthesis, think about it, dehydrating something. Okay, if you're dehydrating jerky, you're removing the water from the meat. In this case, we are removing water from our molecule that we're hooking together. Synthesis means to make something new. In this case, I'm making a new molecule by hooking together these other 2 substrates. Like photosynthesis is making glucose from sunlight, or protein synthesis is making proteins through reading DNA, transcription, and translation. So synthesis means to make something. Dehydration, remove water. I'm removing water to hook 2 things together. So you remove water and you hook 2 things together. Dehydration synthesis. And then we end up forming a covalent bond. If the two things I'm hooking together here are proteins, that bond is actually called a peptide bond. If it was carbohydrates, it's just a covalent bond. That's all you'd call it. But the beautiful thing about anatomy and living things and enzymes is that they are recycled. They don't get used up. Just like your key, every time you unlock your door, it doesn't mean you have to go get and make a new key for your door. That same key works on that same lock over and over and over again. Just like this enzyme gets recycled for the very next reaction. So we never use our enzymes up. We just don't want to ever denature them by getting too hot, by getting something like a fever, something Mrs. Lloyd had early last week. So it's important that fevers don't get too high. And we'll talk more about why we have fevers, if they're bad for us, like why does our body do that to fight off viral infection. We'll talk about that in our immunity unit. So many things we get to learn this year, you guys. All right, so now we're going to move on to page 3 and looking at the top part of this page on the carbohydrates. So you guys already have carbohydrates, monomer, monosaccharides. Saccharide means sugar, mono 1. So our single subunit, the thing that builds up our carbohydrates, that's what a monomer is. The small thing that builds up the big thing is a simple sugar or a single sugar. Now, think of your carbs as your fuel. I'm sorry, guys. I want to go back and mention one thing before I forget about proteins. Okay. One thing I want you to pay attention to on all of the molecules that we talk about today is what atoms we find in them. I don't have it written on the page, but it wouldn't be a terrible thing to write down. There's carbon in this, and there's hydrogen. That means it's organic. Cool. What other molecules do you see in here? Well, every single one of them definitely has nitrogen, okay? And they all have oxygen as well. So I know for sure every single protein is going to have carbon, write that down, oxygen, hydrogen, and nitrogen. And then they sometimes have a few other things. Notice this one has sulfur. They don't all have sulfur. Sometimes they have phosphorus, the P, okay? They don't all have that though. And so you can actually see that on this picture. So we have some that have sulfur. We also have some that have phosphorus. Where's one example of that? I think they're actually in these rings here, so it's not pictured. But just so you know, proteins have all of those different molecules. Now, the reason that reminded me is we're getting into the carbohydrates here, and they have carbon, hydrogen, and oxygen, and that's it. They don't have nitrogen, they don't have sulfur, they don't have phosphorus, or any of the other molecules that are common. Just the 3 basics, carbon, hydrogen, and oxygen. The other unique thing that they have is they always have it in this ratio where there are 2 hydrogens for every 1 carbon, and usually every 1 oxygen. That rules a little. I want you to pay attention to that as we go through our formulas of the different molecules here. So on here we have 3 pictures. Okay, this picture down here is showing dehydration synthesis. We're going to label that at the end, but we're mostly focusing on these top 3 pictures right now. So we have monosaccharides, disaccharides, and polysaccharides. Saccharide means sugar. So a monosaccharide is a 1 sugar, a simple sugar, a single sugar. And notice how it's just a single ring. I'll talk more about the structure here in a minute, but this is basically a carbon ring. So I know it just looks like a bunch of hexagons and O's, and I'm telling you that there's carbon and hydrogen on here. I'll show you on another picture how that's true. But we have here a carbon ring. Okay? There's assumably a carbon here, and then oxygens and hydrogens on either side of those straight lines. So this is a single sugar carbon ring. the disaccharide, notice there's two rings. There are two sugars there, di for two sugars. And then we have our polysaccharides. Poly means many. And so there are quite a few carbon rings on here, and it's all branched out in different ways. So let's talk about monosaccharides. They always have 3 to 7 carbons in its ring, as little as 3, as much as 7. Our classic monosaccharide formula you might have heard of before from Bio 1-2 is C6H12O6, and that's good old glucose. It's not just glucose, though. It's actually fructose and galactose as well. But all three of those molecules have the exact same formula, but how the atoms arrange themselves is different. So they have the same chemical formula, but their structural formula, how I would write it on a piece of paper, is different and unique. They call molecules like that isomers, meaning same formula, different structure, like same but different, isomer. But they are all still single sugars. They have one ring with 3 to 7 carbons in them, and that's their formula. Two others that we will talk about, maybe you've heard about, are ribose and dextrose. Both of those are found in RNA and DNA. Ribose's formula is C5H10O5. So again, think of that ratio, 2 hydrogens to 1 carbon and 1 oxygen. That fits the bill, okay? 10 is double of 5. And then deoxyribose is C5H10O4. And so this is where it doesn't always follow that rule necessarily of the oxygen. And it's mostly because deoxyribose has a strong affinity where it's kind of, it bonds in the backbone of DNA. And so we end up with having 2 sides to our DNA rung because of that missing oxygen. Anyways, it has a little bit of a different formula. So those are some monosaccharides. Now, some disaccharides. Here's the formula for the disaccharides. And before you get on to me about this formula, I will come back to it later. If you notice, it's not a perfect, if you're like, okay, 22 is not double 12. It should be H24, C12. I'll explain why in a minute. But 3 classic disaccharides that you should be familiar with are sucrose, lactose, and maltose. Sucrose is table sugar, nice and sweet and tasty. Lactose is the sugar that is in milk, cow's milk, and it gives milk its sweet taste. And then maltose is another sugar that's in some food products that we eat. It's not as sweet that you would think of as sucrose, though. Then we get to polysaccharides. Okay, and for the formula for this one, I've written it, think of it sort of an algebraic equation, if you will. So we have C6H10, again, I know that's not double, I'll explain why, O5, and then in parentheses times some number, meaning it's a polysaccharide. Let's say I had 20 sugar rings, okay, simple sugars in here, I would put a 20 here, and then all of these numbers would change. If I had 2,000 sugars, okay, I'd put 2,000 here where that N is, and then that would change these numbers inside. You don't have to know the formula for polysaccharides. I won't ever ask you to memorize that, but that's how it's determined. Here are 3 classic examples of polysaccharides that we have in our bodies. So we have cellulose, which is in the cell walls of plants. Starch, which is one of Mrs. Lloyd's very favorite foods. Plant storage. Starches are going to be, right, we think of pastas and breads, but potatoes, think of potatoes, they are the root of a plant. To get a potato, you got to dig in the ground and pull out the root of a potato plant to get it. It's storage for sugar for the plant, and then we humans absolutely love to eat them as french fries or baked potatoes or I'm making myself hungry thinking about this. Glycogen is the only polysaccharide that we have in our cells, in animals. It's the only animal starch there is, and it's in our liver. So when we eat excess sugar, we store it up in our livers for times when we have waiting till 1:05 to eat lunch if you're a bee luncher, or whenever your body is going to use that glycogen that's stored in your liver for energy. So those are our three main groups of carbohydrates, but let's talk about how you can form disaccharides and polysaccharides from them. So that's what the picture on the bottom is. Oh, I'm getting ahead of myself. Here's a very blurry picture, I'm sorry, but it shows you over here the different ways that polysaccharides have chains. So this is showing glycogen, and it's kind of this long branching chain. Cellulose in the cell walls of plants is more really kind of tubular, very tough and rigid. It's like a wall, like cell wall. And starch sort of forms sort of this zigzag pattern in the plant cell, like the potato roots of the plant. And so polysaccharides can be long, super duper long, made up of thousands or millions of simple sugars. All right. So I know you do not have a ton of room. I try to keep my labels small and simple so you could fit them in there, but I know this is teeny tiny guy on here, but I cheated and took it out as a whole little snippet here so you could see everything a little bit better. But this is showing the process of building up or breaking down our carbohydrates. So what we have here is glucose and fructose. The green one's glucose and the pink one is fructose. They don't expect you to memorize that. Now don't draw this. Don't write this down. Okay, gross. But before, when I told you that these pictures were simplified versions of the actual structure of the molecule, this is what it actually looks like. If I wrote out all of the C's and H's and O's of glucose by C6H12O6, this is what it would look like. And if I count it out on here, well, you can count it out right now, there are 6 carbons, 6 oxygens, and 12 hydrogens on there. Same with fructose, right? Right here, okay, fructose as well. But you can see that their arrangement is different. Now, this breaks my brain when I have to look at it. So instead, we have these nice simplified carbon rings that we look at. So these little lines right here are representing, whenever there's a ring, you assume that it's always carbon because carbon loves to form rings. Carbon forms 4 covalent bonds with everything. Carbon's a tramp. It gets around. It shares with everyone. It doesn't care who it is. But it needs to make 4 bonds. So right here in this corner is a C. And notice there's 1, 2, 3, 4 lines coming off of it. That's carbon making 4 bonds. And so those lines coming off in sugars, you can assume that there are hydrogens and OHs coming off. There's usually one section that has a little bit of a chain, but it's not super important to, to note on here. So that is the full out version that we will never do until I actually ask you to build one of these sugars in class with our molecular models later. All right, so when I want to make glucose and fructose stick together, I have to remove an OH from glucose and an H from fructose. That is 2 H's and an O, also known as water. So when I do this, I do, once again, same process, dehydration synthesis. I remove a water molecule to hook these 2 molecules together, and they become covalently bonded, and they end up sharing the oxygen that gets left over here on fructose. So they both end up forming a bond to fill out their outer energy levels, and that molecule that is a combination of glucose and fructose is our very favorite sweetener, sucrose. and that's a disaccharide. So that process of forming sucrose is dehydration synthesis. When I eat sugar, my body actually prefers to get the glucose and fructose separate from each other. So my body uses enzymes, the enzyme sucrase, ASE, and that enzyme has very special cup holders just in the perfectly right shape to fit this and this sucrose molecule. And it adds a little energy, and it also is going to insert in a water molecule. So it forces in a water molecule, and breaks it apart. That opposite reaction is called hydrolysis, the opposite of dehydration synthesis. So looking at root words here, that should help you remember it. In this case, instead of dehydrating the sucrose, I'm hydrating it. I'm going to add water in, hydration, hydrolysis, and lysis, to lysis something, means to break it apart. Lysosomes, those are the cell organelles that break stuff down. When a cell lyses, it bursts and dies. So hydrolysis means to break apart by adding water. Right? So adding water and breaking them apart. Dehydration synthesis is removing water to hook two things together. Okay? Two important vocab words for today: dehydration synthesis and hydrolysis. Okay. Here are the formulas for all of these. It's just right above. You don't need to write them down again, but I have them here for reference. Remember how I told you I promised I wasn't going to forget about the reason why this is not 24. It's because it's not double of that C12. So if I were to add these two molecules together, C6H12O6 times 2, I would have 12 carbons, 24 hydrogens, and 12 oxygens. But I have to remove a water, 2 H's and an O. So when I remove 2 H's and an O, it becomes then C12H22O11. The other 2 H's and the other O ended up over here. So that's why this formula doesn't exactly match that double rule I gave you, but it's assuming we account for the water molecules when we think about that rule. So there you have it. All right, you don't have to write this down, but I just wanted to show you the same graphic going the opposite direction. I used very clever clip art here to change the direction of my arrows. So let's say I have sucrose as my molecule represented right here. Okay, the purple enzyme in this case, is going to be sucrase. Okay, it breaks apart sucrose. It ends in ASE. It's an enzyme. So sucrase fits into those perfect little cup holders. I'm going to add water. So this process where I break apart sucrase is called hydrolysis. Right? And so in hydrolysis, when I break apart sucrose by adding water, I end up with glucose and fructose as my products at the end. Okay. So it works both ways, and enzymes participate in both ways. And so that's why you see on this formula here, the arrows going both directions, okay, is because it's a reaction that can go both ways. All right. Now we're going to check out the bottom of page 3 here and talk about the lipids. So hopefully you've separated those carbohydrates from this because it's totally different topic we're on here. So our lipids, okay, we have 3 main groups of lipids. We have the fats, okay, these are the things that are in on the delicious things. Egg yolks are fatty, also butter, margarine, all our oils, vegetable oils, canola oil, everything fried, it's all in fat. Phospholipids, which we do eat, but you don't really think about it as food. These are the cell membranes of everything you eat. So whenever you eat a plant, you're eating its cell membranes. Whenever you eat other animal matter, meat, chicken, whatever, you're eating cell membranes. You're eating some of these, but your body creates them as well. And then the last group looks totally different from the other two. these are your steroids. And they are going to be your hormones that are found in free-floating around in your bloodstream. So they're a lot different from the other two types. So the structure of these are very different, which we won't really get into. But all of these kind of have a same basic rule. They are made up of this thing called glycerol and fatty acids. You can consider these two things the subunits of our fats, okay, or our lipids, I should say. So when we want to take a molecule of glycerol and hook it to fatty acids, we take 3 of them, and look at this. They all have on them H's and OH's. Actually, do you recognize this? A COOH? That would be a carboxyl group. Throw back to those amino acids. They had carboxyl groups as well. We're going to actually strip the OH off of that carboxyl group in order to hook them together. Right? So when we remove an H here and an OH here and an H here and an OH here and an H here and an OH here, that is 3 water molecules, right, that I've removed. And this exact same process, this little arrow right here is representing dehydration synthesis. So what I end up with as my product is a fat and 3 water molecules. One thing I want to point out are these little dots right here. These fatty acids are really long, much longer than we could fit on our paper, and so those little dots are just saying like this formula continues for a long ways. Now, phospholipids are very similar. They have glycerol and they have 2 fatty acids, but then they also have this unique group here, sometimes called the phosphate group. It's the phosphate group plus another molecule attached to it. It's not important, but it's oftentimes referred to as like the phosphate group or the phosphate head or the polar head, because phosphate groups have a polar charge to them and they are attracted to water. And then they have this little neck region, which is the glycerol. And because of this head region, there's not a 3rd fatty acid here. Instead, we only have room for 2 to hook on. And so there's 2 fatty acids. Oftentimes we call this the fatty acid tails, because we oftentimes think of our, I'm getting ahead of myself here, we think of our phospholipids in this form here. You might have seen this picture before from, again, Bio 1-2. You've got the head, the polar head, and then the fatty acid tails here. And so this is represented in it chemically. Now look at the differences between these two chemically. Again, we have the dots saying like this chain. It continues for a long time. But they are, in this first one, okay, it's just this long chain of carbons, and then coming off of each side of it are 2 hydrogens. And then it's attached to another carbon and 2 hydrogens, going all the way down. This is considered saturated, meaning it's full of hydrogens. If I am saturated with, like my towel is saturated with water, it's totally wet, sopped up everything, the last little bit it can sop up. These fatty acids have sopped up as many hydrogens as possible. They are saturated, or they are full. Unsaturated fatty acids tend to have these have these double bonds in them. And because they have a double bond, remember carbon forms 4 bonds, it ends up dropping some of the hydrogens. So it only has 1 hydrogen instead of 2, because it double bonds with this other carbon next to it. And you can have multiple double bonds in a chain, and these are called unsaturated. And what it ends up doing to the fatty acid is it ends up causing these little bends that you can see in there, these little like kinks in it. And unsaturated fatty acids end up having that characteristic. And so we have, you can have fats up here that are saturated. In fact, these are all saturated fats pictured up above. Or you could also have unsaturated fats where there's a double bond in there as well. And the other thing to remember, okay, with our phospholipids is the head is hydrophilic, meaning it loves water. The tails are hydrophobic. And because those heads are hydrophilic, because they are polar, polar means that it loves water, right? It's attracted to water. Hydrophobic means water-fearing. Just like fats here in general don't mix well with water. If you've ever tried to mix oil in water, right, or even try to wash oil off of your skin, you can't do it with just water. Just water running over your skin with oil on it isn't going to work. You need to add soap in order to break up that lipid layer between you and your skin because it's hydrophobic, water-fearing. It doesn't mix or react with water. Okay, those are our two general ones of our lipid group, but steroids do consist of this group as well. They are, in general, non-polar. They don't mix with water. They have some cool features that they can do because of that in our bodies. Cholesterol is the one that's pictured here. I could show you testosterone or estrol or other steroids as well that we make in our body. But another important steroid is vitamin D. And vitamin D, we will talk about a lot this year. It is an important steroid that our skin actually makes, but you have to get UV rays from the sun in order to produce vitamin D. You can't eat it. It's not like a vitamin supplement that you can take. Your skin actually has to make it, and the only way it synthesizes it is using rays from the sun. The reason why we need vitamin D is it's really important actually for our bone growth. And if you are lacking vitamin D, your bones won't grow the way they're supposed to. There's a disease that can occur in children who are left in the dark called rickets. And rickets is essentially from a lack of sunlight causing the child to have a lack of vitamin D, and it affects their bone growth. So vitamin D is really important. We're going to talk about it when we do the integumentary system and the skeletal system. I believe also when we do our endocrine system and our hormones as well. All right, so those are our lipids. Okay. We aren't going to get into the nucleic acids just yet, but actually, let me go back. We already talked about in the proteins, what molecules you find in them, carbohydrates, carbon, hydrogen, and oxygen, but in that specific ratio. And then I want you to look at these molecules here. What molecules do you see abundantly is carbon and hydrogen, tons of them. And I mean, these chains of lipids are long. And so there's tons of carbon and hydrogens and like 3 oxygens. So very, very little oxygen, but there is still some. So this one does have CH and O, but just a small amount of O, unlike with our carbohydrates where our carbon and oxygen are about equal amounts. So those are the molecules that we find in them. I want to be sure and mention that to you. Then, lastly here, this gets into what your exit ticket is, and I hope you were paying close enough attention to know the difference between dehydration synthesis and hydrolysis. I will ask your guest teacher to ask you that on your way out, and then that's the first thing I'm going to see when I come back on Monday is see if did you get it, okay, the difference between these two. So I want you to explain in your own words the difference between those two. If you want to draw a diagram of them, you sure can, but whenever you draw a diagram, be sure you add labels to help explain the concept. Now, for the rest of the class period, you guys are going to get on the computers. You can use the desktops in the classroom. And if for whatever reason, well, we'll get into that later, but I think there should be enough for every student to use one. And if we run out of computers, talk to the guest teacher. There's a game plan for that. But what you're going to do is get into Google Classroom. And what you're going to do is be assigned from this guest teacher your specific amino acid. And you're going to do just perusing the internet, okay? You don't have to be so, I'm not going to direct you to certain websites, but you're going to research your amino acid. And you're going to look at a few specific things or unique things that it has. So What you're going to end up creating, though, is a Google slide, and then you're going to turn that in Google Classroom for your amino acid research. Even you students at home, I'd like you to do this as well. I will personally message you and let you know what amino acid you have to research. But that amino acid, you're going to get to know it really well. And next week, we're going to build them three-dimensionally with molecules as well. So you'll get to do some hands-on stuff with them as well. All right, so that's the game plan for today. You do have a homework assignment over the macromolecules, so be sure and do the in-class research, but also your homework assignment that's in Google Classroom. Have an awesome day. It was nice to be with you today. See you.